A very warm welcome to all of you guys. In this video, we will going to talk about Neuraminidase, another glycoproteins present on the surface of the viral envelope influenza. Okay, so this enzyme helps virus to be released after budding from the plasma membrane of the host cell. Okay, that means we have talked about hemagglutinin, and during that discussion we have talked about up to that. Then there will be synthesis of the mRNA synthesis, RNA replication, then the genome is ready, proteins are also ready. Now after the budding there will be release of the virus. Okay, and during that procedure there is a problem that occurs, and that problem solved by the neuraminidase. Okay, so we all remember about the hemagglutinin, right? That is present on the surface of the viron, is needed for infection, and its presence inhibits the release of particle after budding. That is the bad thing. While hemagglutinin allows virus to enter the host cell, the same hemagglutinin inhibits the virus from released from the host cell. Okay, why? Because remember, virus have entered inside the host cell due to the sialic acid receptors, right? So after the budding process there is a chance that the newly synthesized viruses hemagglutinins again interact with the sialic acid receptors present on the host cell. Okay, and as a result the virus cannot release or cannot removed from the infected host cell. Okay, and that is why virus have developed the neuraminidase. This neuraminidase have the ability to cleave the terminal cyanic acid residue from the glycan structure on the surface of the infected cell. Okay, and this promotes the virus release of the progeny virus. Okay, so this promotes the release of the progeny virus and the spread of the virus from the host cell to uninfected surrounding cell. That is the idea. I hope it is very clear to you that when the hemagglutinin plays an important role in the entry of the virus by interacting with sialic acid receptors. Similarly, after the development of new virus, the newly developed hemagglutinin also have the ability to interact with sialic acid of the infected host cell. Okay, and as a result the virus cannot removed from the host cell. Okay, under such circumstances, after budding from the host cell's plasma membrane, for ensuring the release of the new progeny virus has developed a new type of glycoproteins called neuraminidase. Okay, which is an enzyme that have the ability to cleave sialic acid from the glycan structure. Okay, and as a result now hemagglutinin cannot recognize it, and it ensures the release of the progeny virus. Okay, not only that, let's say there is a hemagglutinin present. Okay, and let's say somehow there is a sialic acid present in the viral proteins. Okay, that can prevent the aggregation of the virus, and this is also prevented by neuraminidase. Okay, so in one word, what is the function of neuraminidase? The function of neuraminidase is to cleave the sialic acid. Okay, and ensure the release of the progeny virus, and it also prevents the aggregation of the virus. Okay, that is the idea of neuraminidase. And how they are doing this? They are doing this by cleaving the sialic acid structure from the glycan in the host cell receptor. Okay, and remember, neuraminidase activates after budding from the plasma membrane of host cell. Okay, so that is all about neuraminidase. Okay, so we have completed hemagglutinin and neuraminidase structure very detailedly. Now we will talk about the life cycle of the life cycle of the influenza virus later, because it is now not in our syllabus, but I will also made a video on influenza virus life cycles. Don't worry about this, but Now it is time to focus on the symptoms of the influenza virus. Okay, then we will talk about the treatment and diagnosis. Okay, and now let's say for a quick recap, let's again revise about the hemagglutinin. So hemagglutinin is the first type of receptor present on the viral envelope that ensures entry of the virus, right, by interacting with the host cell receptor protein containing polysaccharide terminating with sialic acid. Okay, so this hemagglutinin is a trimeric complex, right? So each of the subunit consists of two things. One is binding domain, another one is fusion peptide. Okay, so how this fusion peptide freeze? This fusion peptide freeze due to the cleavage of the specific portion of the hemagglutinin by a enzyme coming from the epithelial lining of the human respiratory tract, and that activates influenza virus. Okay, now they will enters inside the host cell, and as they are inserting via receptor mediated endocytosis, so they will be covered by the vesicle. Okay, now there will be fusion of the vesicles with the lysosomes. Okay, and this lysosome consisting of acidic interiors and digestive enzyme. Okay, now this acidic interior will be used by the virus itself for their own advantage. They will take all the protons by their M2 ion channel, and they will use those protons to disrupt the protein-protein interaction. Okay, and that will uncoat, that will begin the uncoating process. Okay, then due to this low pH, there will be a conformational changes in the hemagglutinin. Okay, and as a result, the receptor binding domain which was in interaction with the sialic acid will move back, and the fusion peptide which now exposed will direct it towards the vesicle membrane. Okay, now many hemagglutinin molecules together with their fusion peptide try to fuse with the vesicle membrane, and due to this fusion mRNAs are released. Okay, although it will be not mRNA because it is negative sense RNA, so let's talk about only RNA. Okay, the complementary mRNA, that will be more precise word, right? So the complementary RNA will be released into the cytoplasm, right? And that is all about hemagglutinin. And during the recognition of with the sialic acid receptor, they mainly recognizes a linkage between the sialic acid and the galactose. If it is alpha 2,3 linked linkage, then this linkage will be recognized by H5N1 strain, and if it is a alpha 2,6 linkage, then this linkage will be recognized by influenza. So that is all about hemagglutinin. And what neuraminidase are doing? Neuraminidase is another glycoprotein present on the surface of the viral envelope, and this neuraminidase ensures the release of the newly developed progeny virus after budding from the host cell's plasma membrane. And how they are doing this? Because remember, due to the presence of sialic acid on the host cell, there can be a chance that newly synthesized neuraminidase again react with those sialic acid. Okay, for preventing this chance, neuraminidase cleaves the sialic acid from the glycan structure. Okay, not only that, by cleaving sialic acid, they also prevents the chance of virus aggregation. Okay, that is also a very important thing. Okay, so that's all about the hemagglutinin neuraminidase. So I hope these two videos will be very helpful to you to understand about the hemagglutinin and neuraminidase. So thank you for listening to this class.